All right. Thank you so much to the organizers for putting this together. I'm just absolutely delighted to be here, and I've learned so much already, and I can't wait to hear the rest. This is mostly work by Erwin Youngreis in my lab, who's been doing comparative genomics for a long time on mostly mammalian genomes, and Rachel Silfen, who has been working with us for also many years, and who has applied these methods to viral genomes in collaboration with Perdisa Betty. And in this particular presentation, I'm going to tell you some of the results of applying some of our comparative genomics methods to SARS-CoV-2, and specifically how we can use that to revise the gene content of SARS-CoV-2 and to understand the mutational impact of mutations in the current COVID-19 pandemic using 44 sarbecovirus genomes. So first of all, the human genome that we're used to studying is ginormous. It's like 3.2 billion nucleotides. This is the entire SARS-CoV-2 genome. It's 29,903 nucleotides of trouble packed in a single positive strand RNA. And within this lie all of the known genes for SARS-CoV-2, which I'm showing here in different colors. You can see ORF1AB that starts with an ATG, ends in a stop codon, and encodes up to 16 different polypeptides, all cut from a single translational product. And then you can also see the spike protein that's been all over the news, and the membrane protein, the envelope protein, and all of the annotations. So you think there isn't much room to hide either new genes or any much room for mistake for existing genes. We basically know this genome by heart, right? And you could even, I'm sure, just like you can memorize digits of pi, I'm sure there will be competitions for reciting every single nucleotide of SARS-CoV-2. But in fact, in addition to all of the proteins that we kind of know about, there's a lot of ambiguity in the genome. So you know that the genome basically produces both this giant RNA that is the first to be translated. There's a little bit of an echo. Hello? And it also produces these subgenomic transcripts that allow it to effectively translate the rest of the proteins with a single translation product for each of those transcripts of the subgenomic RNAs. But how many proteins are actually produced? Well, here's the gene annotation, and it varies greatly. If you look at NCBI genes or UniProt genes or the mapped genes, you basically see that the actual number varies a little bit, and that there's a lot of variation beyond the known genes. So as I showed on the first slide, we know about the surface glycoprotein, the spike, envelope, membrane, nucleocapsid, but there's a large number of other genes that vary greatly. And what we've done is basically realize that some of them are actually not protein coding genes. How did we do that? We basically used comparative genomics methods that we've developed and applied for many years now, in mammals primarily, but also in fly, in yeast, and in viruses, as I mentioned earlier. And those methods are allowing us to look at an evolutionary tree of multiple related species, and then build alignments for those trees, and then start visualizing and quantifying probabilistically what is the probability with which each of these alignments was generated according to a protein coding model or a non-coding model. Basically because protein coding regions have distinct evolutionary signatures, which are very different from non-coding regions. And if you look at the evolutionary conservation of these regions, you can see a huge, very positive signal for protein coding regions and a huge, very negative signal for non-coding regions, and the two transition quite sharply between them. We've also used Fresco, a tool developed by Rachel Silfen, based on work initially done by Mike Lin in my group, to basically look at synonymous changes, namely nucleotide changes within protein coding regions that change the nucleotide, but have an additional overlapping function within those. And then we use those two tools to distinguish both protein coding regions and to annotate the recent mutations of SARS-CoV-2 during the COVID-19 pandemic. The first challenge was to select species at the quote unquote right evolutionary distance, neither too far nor too close. And we've heard a lot of presentations about all of the species that infect humans. So these are the little stars here. These are basically the common cold that nobody worries too much about. And then MERS, and inside here, this tiny little tree of beta coronaviruses and these sarbecoviruses, so SARS-related beta coronaviruses, as you heard previously, lie this whole group of species, one of which is SARS-CoV-1, which caused the 2003 pandemic, and the second one is SARS-CoV-2. And you can see that even those two are in very different sublineages of the sarbecovirus tree. And these species are basically sufficiently close to be alignable and informative, but sufficiently far to be highly informative about protein coding alterations. So we basically built an alignment of these species, and then we scanned the entire genome to look for these protein coding signals. And these protein coding signals are based on the fact that in protein coding regions, we see a large number of synonymous and conservative substitutions, but in non-coding regions, we see a large number of radical changes, as well as stop codons here, shown in different colors, and frame shifts shown in orange. When we apply these methods, we see this amazingly clean signal for the first ORF that switches reading frame exactly at the frame shift that translates the second half of ORF1AB proteins, and then sort of switching back into each of these different proteins. So that's all nice and good. There's very strong phylo-CSF signal in all of the named proteins. But what about the unnamed ones? So let's focus on the second half of the genome. So we can see, actually, this is the first half of the genome, NSP 1, 2, 3, 4, all of them, very clear protein coding constraint. Second half of the genome, you basically see that there's, for example, no synonymous constraint in the ORF3B, which is predicted to overlap 3A. There is no nucleotide conservation at all in ORF8. So you would say, wow, maybe that region is non-functional. But in fact, you see very clear pockets of phylo-CSF signal, this comparative genomics approach that we're using to detect protein coding constraint rather than just overall nucleotide constraint. And then if you look within these overlapping ORF for ORF14 that overlaps the nucleocapsid, it actually shows no synonymous constraint according to Rachel's methods, suggesting that this is in fact not a protein coding gene. And then if you look at ORF10, you see that there's no phylo-CSF signal whatsoever, suggesting that in fact ORF10 is just not an ORF. It's not a protein coding gene at all. It's probably just part of the terminal RNA structure. So this is in summary. Let's now dive into each of the details. And then the other comment is that phylo-CSF score for ORF6 is, relatively weak and present mostly in the first half. And nucleotide conservation is extremely low. And yet we believe that ORF6 is protein coding. And lastly, there's a new ORF, 3C, which is lurking in the first half of 3A. And I'm gonna, again, dive into that in more detail. So let's dive in through each of those examples in detail and look at their alignments. So this is ORF6. You can see it's a very small ORF. Here's the start codon, here's the stop codon. You can see all of the conservation across the different sarbecoviruses. And you can see that, again, the first half is extremely cleanly protein coding. And the second half appears to have a pocket here of rapid evolution, but then is clearly protein coding as well. So ORF6 is indeed small. It is fast evolving. Its function is not very well known, not very well characterized, but it does make a functional protein. What about ORF8? So ORF8, if you look at the nucleotide level conservation in the UCSC genome browser, you basically see almost no nucleotide level conservation at all. So if you stared at this region naively, you would say, well, that's clearly non-functional. But if you look at the protein coding conservation, you see these pockets of very strong phylo-CSF signal indicate, and no stop codons over the entire length, and very good start and stop conservation, indicating that it is indeed protein coding, even though it is very fast evolving. So it's as fast evolving or even faster than the S protein. So for those of you interested in figuring out sarbecovirus biology adaptation and how it's so successful, I think ORF8 is a very good candidate for functional studies. ORF8 also pseudogenized within the SARS-CoV-1 population. It basically was split into ORF8A and ORF8B, and that actually altered its transmission. It made it actually less transmissible, suggesting that ORF8 might actually be functional in transmission in virulence. Here's ORF10. It's at the end of the genome. So you see this, very clean signal for 6, 7A, 7B, 8, N, and then 10 is at the end. Let's see if 10 is real. So what do we see? We basically see a start codon here. You see a stop codon here. That's all nice and good. But then you also see a premature stop codon in most of the genomes. And you see that these changes don't look like the lime green that I was showing you earlier. There's basically mostly red changes when there are any changes. And the other thing that should be very worrisome to all of you is that there's a lot of white. What does white mean? White means there's no mutations. So a protein coding gene usually shows mutations. This does not show mutations. So we believe that it's very highly conserved at the nucleotide level rather than at the protein level. And that's the reason why the start and the stop appear to be conserved rather than the fact that they are, a start codon of a protein, which they're not. So we differ in this conclusion from recent papers by Cagliani and Finkel, which basically provided evidence that ORF10 is functional. But in fact, that evidence is itself a little controversial. So basically, Cagliani excluded a frame-shifting deletion in one of the strains. It only used 6 strains instead of the 44 strains that we're looking at here. And in one of them, there was a frame-shifting mutation. And that one was deemed to be possibly a sequencing error, so it was not analyzed in detail. And Finkel also reported footprints for ORF10, but if you look at where are the translational footprints, they are before that ORF or after that ORF, but not quite within the unique portion of ORF10. So that suggests that ORF10 is probably not a protein coding gene. So we then turn our attention to the dual protein coding regions. So basically, if you look within the nucleocapsid ORF, there are 2 proposed overlapping ORFs, 9B and 14. 14 looks really terrible. It's unlikely to be real, and it has no synonymous constraint. So we believe that 14 is not a protein coding gene. And then ORF9B actually does show some signal of conservation. It does show some overlapping constraint. So we believe that 9B is ambiguous, but more likely to be real than not real. Whereas 14 is quite unambiguously not protein coding. ORF3B has been proposed as an overlapping ORF that spans the end of 3A and the beginning of E. And we believe that it is not protein coding. If you look here, there's a stop codon, there's a few internal stop codons, and the overall conservation pattern looks extremely non-protein coding. So it's unlikely to be real. Some authors have proposed another ORF that they call ORF3B. We call it ORF3B2 for lack of ambiguity. And indeed, this has a lot of premature stop codons, and it has a lot of non-conservative changes, indicating that, in fact, ORF3B2 is also not real. What about novel genes? So we then looked at the entire genome for all ORFs that are more than 25 codons and scored every single one such interval to basically look for the protein coding conservation signal. And what we found is that all of the known proteins are here. Some of the overlapping proteins are here. So if you look at 9B, which is overlapping, you see that it has a negative score, but that's expected, just like for 10. And the reason for that is that when you have protein coding constraint on one frame, it actually influences the constraint of the other frame. But there was one surprise here, which we call ORF3C, which is very seemingly protein coding. If you look at it, you basically see that ORF3C is encoded in the first half of 3A in an alternate reading frame. And you can see this overwhelming signal for synonymous constraint on one hand, but also for substitutions that are conservative within ORF3A. And in fact, that signal is even stronger than the signal that you see for normal ORF3A. ORF3C appears to be dominating in that part of the sequence. And this was again reported as a candidate ORF in 2 additional papers. So we're not the first to report this, but we believe we have the strongest and most conclusive evidence by far. So that's the summary. We basically have rejected 1, 2, 3, 4, 5 genes. We have accepted one, and then of course confirmed all the others and proposed a new one here. We then turned to the mutations, and D614G has been all over the news, but it's not the only mutation happening in the current pandemic. the first thing we asked is, how are the mutations in the current pandemic mirroring the evolutionary rate that we see across coronavirus strains? And what we see is that indeed the slow evolving proteins across species are also slow evolving in the current pandemic, and the fast one across species are fast evolving in the current pandemic. There are 3 exceptions. Nucleocapsid appears to be faster evolving in the current pandemic than you would have expected across different genomes. But NSP3 and most notably S1 appear to be slower evolving in the current pandemic than you would have expected. So S1 is very rapidly changing between strains, but very slow in the current pandemic, as if it was either slowed down somehow or pre-adapted for human transmission prior to the initial sequencing of the current genome, or if the fraction of amino acid compositions is actually different between S1 and the other proteins that lead it to have fewer of these sort of rapid evolution changes, or if there's simply cycles of adaptation where S1 as a receptor is the first to evolve, followed by adaptation of the other proteins, and that's what we're experiencing. At the level of individual nucleotides, you see a lot of agreement between sarbecovirus conservation and SARS-CoV-2 variants, with lower missense density of nucleotide variants in conserved amino acids, higher missense density at the 3' end of the genome, lower synonymous changes in synonymous constrained regions. And as I mentioned earlier, nucleocapsid is very fast evolving in the current pandemic, and you see indeed an island of many amino acid changing variants within deeply conserved amino acids. As I promised, we also looked at D614G, and this is quite striking. That mutation has never been made in the equivalent of 60 million years of evolution if you were looking at mammalian clocks. So this is an extremely conserved nucleotide in an extremely well-conserved region of S1, indicating that this is unique to human host adaptation. And we also evaluated all of the mutations found recently. So there's a lot of resources out there. We've put our paper on bioRxiv. We've made the alignments available. We have phylo-CSF track. We have synonymous constraint track. We have Caudaline view for everything. And there's a track hub. And as I mentioned earlier, Erwin Youngreis led this work in collaboration with Rachel Silfen, both of them in my both former students. And we've had a lot of very nice conversations with several folks. And the SNVs are, of course, thanks to NextStrain and GISID. And I'll stop there and take any questions. Thank you.
Okay, and it looks like we have some questions from, I think, Thomas Gingaris. Have you looked at the homology and the predicted non-coding RNAs with human genomes, especially ribosomal RNA genes?
Oh, this is a fascinating question, Tom. Thank you so much. Homology of predicted non-coding RNAs with human genomes, that's fascinating. So no, we have not. And indeed, you know, obviously the virus is exploiting the host machinery. So any kind of homology there would be very interesting. But Tom, something that you would be very interested in is the fact that there's a lot of these synonymous constrained nucleotides that are likely overlapping non-coding functional regions, and that are overlapping additional RNA structures. So as you know, this sort of swapping of leading end in all of the reverse transcription of the subgenomic RNAs is likely to have a lot of RNA structure there. And we're also finding a lot of synonymously constrained regions within those protein genes that actually might be binding sites for different RNA binding proteins. So there's a lot of potential interest there.
Pardon me, I do not see any further questions. There is one other question. There we go, from Ian Wickersham. Do you think the rapid mutation rate of N is due to the immune pressure in humans? And are there any mutations similar to those found in native human coronaviruses?
Oh, that's fascinating. So yeah, we have not looked at that, and that's a great suggestion. So basically what you're suggesting is to go back to the other human adapted common cold, you know, SARS-CoV-1, MERS, and to basically see if there might in fact be convergent sites of change or even just increased number of changes within the same segment of N associated with human adaptation. So it's a fascinating suggestion. We'll go and do that. As for immune pressure in humans, so the nucleocapsid protein is obviously holding together the RNA, and that's extremely important in multiple life stages of the virus, in the packaging, in the protection, in sort of lack of denaturing, possibly in the reverse transcription. So it's unclear how it interacts with the human immune system. But if you have information about that, I would love to chat offline. So Ian, please do email me. And then from George Pavlakis, he's asking, what do you think accounts for the rapid change of nucleocapsid? This is paradoxical. Indeed, it is paradoxical. And if you look at the changes, this is the only genome-wide significant change in the recent pandemic throughout the entire genome. So it is both fast evolving in terms of the total number of single nucleotide variants, and moreover, these variants are not randomly distributed, but they're heavily clustered in that segment. So I would love to talk with you, George, about that, and also about Barbara, who's the one posting the question. So yeah, it does seem awkward. This is not the protein we would have expected to be fast evolving at all. I mean, our bets were on S1, but in fact, S1 is much slower evolving, and N is much faster evolving. So I don't know, a lot of mysteries there, and I would love to sort of interact with you guys to learn more about this.
Okay, and there's one more question in the chat box that if I can get Philip Davis to post that on the Slack, then that's a good place for you to answer it there. I don't know, Eric, would you like a final word or perhaps a plug of NITO 2021 before we wrap up?
Well, I can put it on the wall again. if I move over a bit. It is difficult pointing. You're right. So this is the new website.